I'm recovering from a bit of a throat cold, so I might take a sip of water every now and again. There's so much to discuss. Thank you so much for inviting and allowing me to be a part of this. I'm a basic researcher by training who then became a genetic counselor who now works on Simon's Searchlight for the last several years. And so I thought that it would be great, and speaking with the organizers, to have a little bit of a refresher, not deep dive. There's really excellent speakers here today that will definitely give more background and insight, but just a little refresher on basic genetics, sort of show some of the GRIN variants that we have, talk about the GRIN proteins in the brain on the whole and some of the terminology that might be coming up in these talks, and then obviously Simon's Searchlight. So Simon's Searchlight, really wanting to get across how we work with families and how we can work with researchers. So on the whole, the GRIN-related disorders are, sorry, I'll be using this pointer. On the whole, the GRIN-related disorders obviously are related, and they're really important for creating subunits of the NMDA receptor. So what that just means, and in the basic genetics steps, you have the DNA, so the GRIN gene, within the nucleus of the cell. And if anyone is unfamiliar with this, I picture it sort of like an egg, and it's in the yolk. That's where the DNA stays. Most cells have DNA, not all of them do, but the majority of them do, and they would have this genetic variant in all of these cells that have a nucleus. There's a molecule called RNA that is read off this DNA. That's like the next step. The DNA sits in the cell, the DNA becomes this RNA that's read off of it. But what really ends up being important is this protein. And a little bit confusing in GRIN disorders, the DNA name and the protein names are different. And so we'll get to what the protein names are. But what you might have seen on your genetics lab report is, for example, this GRIN1 and a C dot, which means your DNA coding sequence. This just shows that in this reference genome, this site 1607, this A in the reference genome, the usual spot where there's an A, it became a G. And so this small change, usually that happens spontaneously, changes this ultimate structure of this protein. It changes, in this particular example, it changes one amino acid. So the protein has a different letter code than the DNA, and the P dot is the equivalent protein location of this genetic variation, this genetic change. So at site 536, where in the reference it's usually a Q, now it's changed it to an R. And so what that means for this protein is different for every single amino acid change. It could be changing the way this protein locates in the cell, where it should end up. It could change how it binds to its binding partners. It could change how it anchors the substrate or accepts the substrate, which I'll talk about what those are for the GRIN-related disorders. It could do all three. It could do more. It could do only one of those. So that's why you'll see that it's really important with every single variant to study and understand what they're doing, because it's so different in every single genetic change. For Simon's Searchlight, we have 4 of the GRINs on our genetic registry right now. So GRIN1, GRIN2A, GRIN2B, and GRIN2D. This is not our most updated list. Feel free to take a photo. But this is what we're finding, and we're including the variant classification for all of these. I'll show the GRIN2B. GRIN2D, we only have four genetic lab reports, and we need at least 5 for a data release. But you can see that some of the genetic changes that are associated with the condition are happening in certain regions. For GRIN2B, we have so many more variations, so I made a quick image just so that you can see these regions where that commonly is just happening, where there's an effect on the genetic variation. But we do have a lot more that I haven't shown just because it doesn't fit in this slide. But I'm happy to share these variants in the upcoming months because we're about to get a data release. So we have this information publicly available once there is a data release. So we'll have a big release of our list of variants upcoming whenever that does happen, but fairly soon. And so now this next step of what this means for the brain. Obviously, the brain is a very complicated and interconnected structure. And I'm only showing here one of the types of brain cells. There's tons of brain cells. There's tons of different types of brain cells. But in this image, you have two neurons, one of the brain cells, and they're touching each other. And it's not as linear as this because there's different regions of the brain. It could be interwoven. But essentially, from the basis of how they communicate, when one neuron fires for me to lift my arm or start speaking, it's connecting with another neuron fairly fast. And the site where they actually interact is called a synapse. That's this blue region. They're not actually touching, they're just really close. As maybe they are touching on a molecular level, they might not be, but maybe they are actually. So this distance isn't super far. But this structure is the synapse, and that's where this communication happens. And what's released when there's a firing of this neuron is these chemicals, and this is what I was calling the substrate. There's many different types of these messengers, these neurotransmitters that happen to get this other neuron to also fire, to react, to respond. And these particular structures here that you see at the receiving end, there's many different types of these acceptors, the receptors, channels that are important, but NMDA receptor is one of them. And that's where the GRIN genes come back in here. Each of the GRIN genes code for one of the NMDA receptor subunits. And the subunits there, it has to come together in a structure of 4. And the combination of how they come together is different in every neuronal cell. It's different in different regions of the brain. And all the genes are important to work together for this to happen. So as you can see here in this top image is there's 2 GRIN1s, a GRIN2B, a GRIN2A, and they're binding. They allow for a binding of what is called a glycine, a molecule called a glutamate. In general, sorry, I'm going to take a sip. It is the GRIN2s that bind the glutamates, and it's the GRIN1s that bind the glycines, but you can make a combination to make an NMDA receptor, but it usually consists, the rule is 2 GRIN1s and 2 GRIN2s, or a combination of 2 and 3. And now I'll come back to what the protein names are, because you'll see a lot of this protein names in the presentations likely. So the GLU-N1 is GRIN1. How you can remember that is this 1 at the end. And then the 2B is GLU-N2B. The 2A, GRIN2A is GLU-N2A. And that's how you can correlate these GRIN genes with the protein product at the end. And I'm just showing an example with GRIN2D, make sure that they're included in how this structure comes together. And this is just another example of how this NMDA receptor could be made at the synapse to receive these molecules. So that was really digging into some of the basic science. And again, there's some experts in here that are way more versed on what that means in the end for the genetic variations. But for the switching now over to the Simon Searchlight Registry, I would love to start by saying I'm one of the representatives, but this is a very large team of individuals. We have our principal investigator, Dr. Wendy Chung, and Dr. Cora Taylor. We have our project manager. We have other genetic counselors. We have research assistants. We have data management teams. So I'm standing here today as just one of many. And it takes a lot of work in order for this project to work properly. So when you're contacting Simon Searchlight, it may not be me, but it might also be me. And for families participating, there are a few ways that I want to express how we interact with you on what you might see on a day-to-day. So from our goals of collecting medical and behavioral information, along with the blood samples, and I can talk about blood samples coming from other countries than the US. It is really tricky, not impossible when there is a specific research need. But can be very difficult and not routinely done. And we do collect from participants all over the world the medical and behavioral information. We also provide and synthesize this information back. We're always evolving how we do that, so I will give more detailed examples. And then for the researcher side, we share this data for free. We do not charge researchers to access our data. We do have a small charge for the IPSCs and biorepository, which I'll also show in an upcoming slide. We make you able to be found by researchers, so researchers can come to our registry and say, can you send my research study to every GRIN family located in Europe, located in Australia, all participants that have a pathogenic variant. We make it so that researchers can contact you and share their research studies with you. But researchers, you can implement research studies and recruit through our registry. And we, again, do not charge for that. We're happy to do this. And then just the ultimate, of course, goal of being a resource for the community, promoting better understanding and access for research in the rare neurodevelopmental space. So how this is all possible is that we're funded through the Simons Foundation. So this is a nonprofit foundation. We are entirely internally funded by this foundation, so we do not take donations or need to collect funding. But we're just one of many research studies under this foundation. So Simon Searchlight being one of them. There's also the Spark research study. This is a US only study where Simon Searchlight is international. And then we have, and the Spark study is for individuals that don't have a genetic diagnosis and have a diagnosis of autism, they get genetic testing. And anyone with a GRIN related disorder gene that's on our list is sent Simon Searchlight information to come and participate in Searchlight. We also have Autism BrainNet, so that's post-mortem brain tissue that's collected, and we run in the US, Canada, and the UK right now. So researchers can also collect and come and request samples from individuals with these rare genetic conditions. The other thing that we have, and it's really important to point out, is funding. So funding for researchers everywhere. We want researchers all around the world applying for our funding. And we often give feedback as to how to make a project stronger. So feel free to ask and find out what funding we have. And then we have other research resources like our animal repository, our Safari Gene online public genetic database. And then we have Safari bases where these requests come in from researchers, where your data is living. So the genetic conditions we study, this is our full list. It's always ever growing and evolving as we learn more. GRIN 2B has been on our list since 2014. And the GRIN 1, 2A, and 2D have been on our list since 2019. And participants can participate in English, Spanish, French, and Dutch. And German and Portuguese and Italian are our next ones that are really high on our list. We're really trying to get it going. The block is that some of the surveys and such that we're trying to get in these other languages are stalled. So until we are able to figure and work through that, which is being worked on, it stalls us to add more, but we really are planning on adding more. That is the intention, to continue to service globally. What we collect, baseline, meaning that the first time you come in, there are a few asks that are sort of bigger. It is a bigger first time investment. And then over time, every year, we want to follow up with you on certain aspects, but we're also changing what we ask as we learn more. So just like we need the genetics lab report, we do medical history. We want to know everything in this medical history, including prenatal history, including intestinal issues and autoimmune issues and asthma. We want to know it all. We're not just focused on autism, and we want to get a complete picture. And we have these other specific surveys about sleep, for example, and seizures and quality of life and such. And there are more that we're adding and planning on adding that we've heard is really important for the community. So that's also being worked on by our team. So for families, what we give back and what you can access right now is available on our website is quarterly reports or registry reports. Every 3 months, we do a different focus. So GRIN 2B, the only requirement for this is that we have 10 or more people with a pathogenic or likely pathogenic variant. And so that way we can give a meaningful result or meaningful summary. So GRIN 2B, we definitely have that number, and we focus on different aspects of what we're collecting in a relatable way. But some doctors have contacted us and said from other countries being like, oh, my patient brought this to me and I want to know more about this, this, and this. So it's really usable and you can access that today. And this is on our public website. You don't have to log in or participate to get this. Whereas we do have some personalized survey summaries where once you're participating, we do a comparison of, for example, this is a survey summary, very basic, but saying, here's where your child was for this survey, and here's where the rest of your community is. So we do some of those personalized summaries if we can for those surveys. And then your information is used in research. So these are just 4 publications as of right now that has used GRIN data in their research. We have 4 other requests of researchers using our data that isn't published yet or is being used. But when a researcher comes to Simon Searchlight, if they say we want SCN2A, we don't clean out the GRIN information. They still get that. So it's a cross comparison. Researchers can access and look at the de-identified data across our whole cohort. I myself do some cross cohort analyses, and there's some real benefit of looking across our different conditions that are connected. And so as mentioned, Research Match is this program that we have where researchers can find you. For the sake of time, I think I covered fairly well at the intro, but I'd be very happy to discuss more with any researchers in the audience. And I don't think I need to sell to this room that working together with genetic variations or genetic conditions that are related and interconnected is really powerful and important to be progressing the community's research and understanding. So for our GRIN-related disorders within Simon Searchlight, I'm showing the 4 here. But what these numbers represent is, as of yesterday when I looked, the total number of individuals that signed up online. So usually this is the highest number because that's the first step, and there's drop off in certain steps. But for individuals that provided a genetics lab report, that's the second row. Then we have the medical history information. We count this as a really important data point because it does the cross comparison. Filling out other surveys, also super important, is another full count of how many people have filled out other surveys, and then the number of blood samples that we've collected for each of our communities. And so, as mentioned, this is where you can access, for example, the GRIN2B data today on our webpage. This is the current genetics lab report. So if you go to simonsearchlight.org, GRIN2B, the current registry report is at the very top. Our previous reports are down here at the very bottom. They're all listed. So please peruse and look through what we found at your leisure. And then finally, a really important point and a really important subset of our study is that once we collect the blood samples, we've been turning some of them into induced pluripotent stem cells. And so for the researchers in the room, we really want you to access this. I believe there's a few more being added to the pipeline, but have not been announced as of yet. All these top 10, 11 are available for request right now. And the cost is as low as $120 per sample plus shipping and handling. We realize shipping and handling can be very expensive still, but we're really not trying to make this cost prohibitive. And so these are available right now. Please come and talk to me if you want to find out more. And the last few points I want to make, because it is of interest to the community, we do follow the guidelines and requirements of GDPR. We are very serious about data protection. So we actually have two databases. So the database where you put in your phone number and your email and your identifying information is different where we house the medical information. So the medical information is de-identified, and that is what gets out to the researchers, not the database that we have that includes your personal information. And so I wanted to touch upon that as well. But thank you so much, and I look forward to questions later.